All right, guys, so today we are going to start talking about how our body makes proteins using the instructions that are found in DNA. So previously we talked about the structure of DNA and the fact that it has 4 different nucleic acids: adenine, thymine, cytosine, and guanine. Now DNA is a very, very large molecule found inside the nucleus. Remember that the nucleus protects DNA, and the only time the nucleus goes away is when your cell is actually dividing. That's when the nucleus degrades in prophase of either mitosis or meiosis. So DNA, because it's such a large double-stranded molecule, cannot just leave the nucleus through these little nuclear pores that are found in there. So somehow you have to be able to get that message from the nucleus to the organelle that actually makes proteins, which in this case are the ribosomes that are found either floating out in your cytoplasm or on your rough ER. So when we learned our organelles, we talked about the fact that ribosomes do make proteins, and I said a little trick to remember that is to think about ribs, and ribs are made of protein because they're meat. Okay, so ribosomes make protein. So we got to get the instructions that are found in the nucleus to the ribosomes so the ribosomes can make the proteins. And the reason why we're talking about proteins so much is when you're talking about body structure, all of our body structure is made out of a variety of proteins, from muscles, skin, ligaments, tendons, hair, everything. It's all different proteins. So it's these proteins that give us our physical characteristics. And DNA is the instructions for how those proteins are made and arranged. So let's jump in to how this works. So the molecule we're going to be talking about that helps this whole process occur is DNA's skinny cousin, RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. RNA, skinny cousin, is only single-stranded, where in DNA you have 2 sides. This one only has 1, so it's half the size of DNA. It's still made out of nucleotides, just one side of them. And the nucleotides are very, very similar to DNA in that you have the little sugar houses, you have the phosphate, and you have the bases. But there's still some differences. So in DNA, the sugar is deoxyribose. In RNA, it's just ribose. So that's why you have a D there and an R there, because RNA is ribonucleic acid. So the first letter has to do with the name of the sugar that's in the molecule. There's also differences in the nitrogen bases, where DNA has A, T, C, and G, right? RNA has A, U, C, and G. So there's this new molecule here called uracil. It's just chemically a little bit different than thymine. It's not a huge amount different, and I'll show you in a picture. But for some reason, our body evolved having this uracil instead of thymine. Everything else stays the same. But what this means is when we're flipping our codes, if we have an A in our DNA, it's not going to turn into a T in RNAs because RNAs don't have it. It'll turn into a U instead. So that's the one tricky part, that if you practice it, it gets very, very easy, but it is the one big change. So here's our strand of DNA, the double helix. And there's our A, G, C, and T. Remember, these are pyrimidines, and this is purines. And then over here on the RNA side, you can see it's just one side. It's like one side of the DNA. We still have A, G, and C, and then here's uracil. And so if you look closely at uracil, you can see some slight little differences in the groups that are hanging off this carbon ring, and that's what makes it a different chemical. We have 3 different types of RNA that are put to work during the process of making these proteins. The one we will be talking about the most is the messenger RNA, and we abbreviate this as mRNA, lowercase m, capital for the RNA. Messenger RNA does what its name says. It carries the message. So it's actually going to come in and make a copy of the code from the DNA by doing complementary bases. So T to A, C to G, G to C, but A is going to turn into U, okay? So complementary bases, and it's going to carry that message to the ribosome. So that has a very big role in this process. And then we got this one right here. This is transfer RNA, and transfer RNA is pretty easy to identify because it looks like it has a lowercase t here in the middle. Almost always looks like that. And then it has like a little thing hanging off the top. That's an amino acid. If you remember back to when we learned organic compounds, we said amino acids are the monomer of a protein. So it picks up these amino acids from the food you eat. When you eat protein, when you have a steak or a hamburger, picks up those amino acids from the cytoplasm of your cell and carries them to the ribosome. But there's a little code down here, a little 3-letter code that will make it match up to this mRNA here, okay? And all this runs by those codes. And then the last one, which we won't talk about a lot, is ribosomal RNA, or rRNA. This is basically your ribosome. So your ribosome itself is made out of a balled up piece of RNA that makes the proteins. So mRNA, tRNA, and rRNA. So here's basically the central dogma of protein synthesis. This is what has to occur. You go from DNA to RNA, and from the RNA to the protein. And so there's 2 major steps, DNA to the mRNA, and then the mRNA to the protein. And each one of those steps has a name and a process that goes along with it. So the first process is transcription. And as you saw in the video I had you watch for your bell questions, it's transcription and translation, and they sound just alike, but they do go in alphabetical order. So the C1, transcription, comes first. You can also think of this word as transcribing notes, like a secretary might transcribe notes for his or her boss. And when they do that, they're using the same language. If your boss is speaking English, usually you're writing it down in English. It's the same language, you're just making a copy, and you might use some shorthand to shorten it up, but it's mostly the same. Well, this is what we're doing when we're going from DNA into mRNA. We're using the same language of the nitrogen bases. There's some slight differences because of that uracil, but we're using the same language. It's still nitrogen bases. So this occurs in the nucleus because your DNA has to be in the nucleus. So mRNA will be made by enzymes called RNA polymerase. Remember, DNA polymerase made DNA. RNA polymerase is going to make RNA. So RNA polymerase, which is this big blob in the animation, will come in and add complementary bases. And you notice the sugar is red here instead of green, if you're not colorblind. And so that is the ribose sugar. That's why it's a little color different. So it's going to make a copy in this replication bubble here. And it's going to take that code out of the nucleus, because it's half the size of DNA, it could fit through the doorways, and to the ribosome. And that's what's happening in transcription. So steps of transcription, for the details, here's the name of that enzyme, RNA polymerase, and again ends in ASE, and it's making the RNA polymer. So name makes sense. RNA polymerase attaches to your DNA, and it attaches at this site called the promoter. So the promoter is promoting the start of transcription. It says, hey, attach here and copy this gene. So every gene has an area on it, a little code that is your promoter that says start here. DNA is your template. That just means it's the part that's going to get copied. Then your polymerase is going to add those complementary bases to form a new RNA molecule. And it follows that complementary base rule, except where there would be a T, you now have a U. So T still goes to A, but A goes to U. After that, transcription ends at another code called a terminator. So, terminate means to end things. So there's a code there that says, hey, stop copying now because you only want this one gene. And at that point, both the DNA and the RNA are released by the polymerase, and the RNA leaves the nucleus. This is how all kinds of RNA are produced, both mRNA, tRNA, and rRNA, all made the same way. So here's a quick animation of transcription. You can see the complementary bases going in, and you have a U there instead of a T. That green guy in the background, that is RNA polymerase. The blue is your DNA, and the red is your mRNA with that new ribose sugar and uracil. So you can tell typically just by the codes if it's DNA or RNA, because if there's T's in there, it's got to be DNA. If there's U's in there, it's got to be RNA. Okay, so in this picture, we can see this is transcription in real time. So that is in green, your RNA polymerase, and yellow is your mRNA being produced off the DNA, which is this reddish string here. And so in your cells, this is how fast this is going. It's a very fast process. So now let's talk about translation. And if you're taking a foreign language class, then you know that translation is when you're changing languages. Like if I'm taking Spanish, I translate from English to Spanish. Well, we're going to change our language here too. We're going to go from the nitrogen bases to a protein, which is made out of amino acids. So this has to occur at the ribosome. So the mRNA that left the nucleus that we made in transcription is going to go to the ribosome, out and float in the cytoplasm or on your rough ER. And there it's going to meet up with tRNA, which is transfer RNA. It's called transfer because it's transferring something to the mRNA. And what's transferring is amino acids. And these came from the food you ate, the protein. So these amino acids are going to be attached in order according to a code. And they are the monomers of proteins. So the name of the little code, which always is in 3 letters, and we will always read these little codes in 3 letters, is called an anticodon, and there's a little T in there, like tRNA. That's the 3-letter code. So if you look down here real small, these 3 letters down here, that's your anticodon, and that's going to meet up to the complementary bases on the mRNA. So here you can kind of see the orange is my anticodon, this big brown blob is my ribosome, this here with the red is my mRNA, and you can see complementary bases, C's with G's here. And hanging off of them with these little letters, these are my amino acids. And this protein already has 2 sitting here, okay? But there will be more as we go down the strand. So the tRNA anticodon is going to bind to the complementary 3-letter code on the mRNA that we saw in the previous slide. In mRNA, it's called a codon. So you have codon, anticodon, because they're like opposite of each other, but they're complementary bases. All right, so this binding lines up the amino acids, and the amino acids then will stick together. They will bond together, and they bond with what's called a peptide bond. So again, at the beginning of the year, when we learned organic compounds, we talked about proteins being polypeptides. So that's how we get a polypeptide, is having many peptide bonds between all those amino acids. So all these bonds form, and then your protein will detach, and then after that it curls up. So here's blue is the ribosome, the straight line is your mRNA. These little guys that showed up, these are your tRNAs, and see they were doing complementary codes. And then these little guys up here, these are your amino acids that have peptide bonded to each other. And then when this is done, it detaches and it folds up into a particular shape, and then you have a protein. So here's it in real time again. The green weird triangular shaped things are your tRNAs. Red is your protein. Yellow is your mRNA, and this blue thing in the background is your ribosome. So these guys are showing up and attaching to their complementary bases and then releasing their amino acids as they peptide bond together. And then that will coil up at the end and make a protein. All right, and here's just another picture showing that process. Here's my mRNA, long, and it has U's in it. Here's my tRNAs with 3-letter codes on it. That is an anticodon. This is a codon, and they complement each other. A goes with U, C goes with G, right? And then hanging off of them are all these little amino acids that will attach to each other. So proteins are made up of sequences of amino acids. So different kinds of proteins within our bodies have different sequences, and that's what makes them different. There is 20 different kinds of amino acids, and thank goodness we do not need to memorize them, because there's a handy dandy little chart that we can learn how to use, which will tell us which amino acid we're using. So here's the chart that we're going to use right here, and you might be like, how do I use that? Well, notice we have the bases, and these bases have U's in them. So this is not in DNA. This is in RNA. So we use our mRNA code to find our amino acid. And they've abbreviated all the amino acids names with 3 letters, which is nice because the names are quite long and you wouldn't want to write all of that, okay? And you'll notice over here that says first base, second base, third base, right? It's kind of like baseball. First base, second base, third base. So if I had a code, let's say I had a code, let's do AUG is my code on my piece of mRNA. So to do this, what I would look at, let me get my highlighter. The first letter is A. So here's my first letter A. That tells me that it has to be one of these 4 across there, okay, in one of those boxes. My second letter is U. So here's my U, and I go down this way. Well, now the only one they're both in is this one right here. So this right now, this is my box where the 2 lines went together. And so usually I don't draw this out, I just use my fingers and where the 2 come together. So the first 2 letters will give you your box, okay? Now you'll notice within these boxes I have 2 different kinds of amino acids. Sometimes you don't, like this box over here, they're all the same and then you don't even have to do anything else because it has to be LEU if you land up in that box. Or in this one, you only have 1 option, in this one too. But in this one, I have 2 options. So the last letter, let me switch colors. The last letter will tell you which one in that box it is. So we use A and U to find this box. And then you go over here where the third base is, and we have a G. And the G is the last one in the box, which means AUG gives me that. It gives me the last base, so it gives me MET. So let's clear that one off. And let's try another one. Let's say I have, I'm just going to make one up. Let's do CGU. I don't know what that's going to give me. Okay, CGU. So we're going to take my highlighter here. And first letter is C. That's my first base. So here's first base is C. Tells me it's in this row. My second letter, G, second base, here's G, tells me it is in this column. Now, I happen to land up in this box. In this box, they're all the same, so I don't really need to do anything else, but just to show you that last letter, that U, there it is, it's the top one. So it would give me whatever this top base is, which in this case is ARG. So that's how you use your mRNA chart. Now you'll notice that some of these give you what's called a stop. All right, we have a couple stops. These are your terminators. They tell it to stop copying at that point. Don't do it anymore. And your promoter is actually this MET one that I start with. That's the first one that starts the copying. So when we practice on our worksheet, you will see that. All right. So hopefully that clarified for you how to use an mRNA chart. I will post another video in case you need